Today's class we are going to study about pollination. In the last class I have told you about the gametogenesis, that is both microsporogenesis and megasporogenesis. Now we are going to read the next stage, that is how the gametes will be transferred. So let us see this diagram. Now pollination is a process by which the pollen grains are transferred from the anther to the stigma of a flower. Now when it is between the two different plants, suppose this is a A plant and this is a B plant, and there's an agent, say this is a butterfly carrying the pollen from the anther to the stigma of the B plant, then this type of pollination will be called as your cross-pollination. It is also known as your xenogamy. But if the pollination is done within the same flower, but of course the flower has to be bisexual, that is both the male and the female reproductive part should be present. In that case, that will lead to self-pollination or autogamy. So depending upon whether the pollination is happening between two different flowers of two different plants or within the same flower, pollination can be classified further into two, that is cross-pollination and self-pollination. Now there are in nature self-pollination happening in the same plant between the two different flowers but of the same plant. So what do you call it? So that type of pollination in which the flower is in different branch but of the same plant, that should be also genetically similar or genetically identical. So that type of pollination also will be, you can say it to be a kind of self-pollination, but it has a different term. We call it as your geitonogamy. So pollination, cross-pollination between two different flowers of two different plants, self-pollination, same flower, and also self-pollination in the same plant but in the two different branches. Now agents of pollination. Now there are, depending upon whether the agent is biotic or abiotic, the pollination is of various types. Now biotic agents will include your insects, it can include your birds, it can include your small rodents, sometimes snail, sometimes human beings when they are doing the programming of artificial hybridization. So in that case they all will be under this biotic agents. When it is done by insects it is called entomophily. When by means of birds it is called ornithophily. So likewise there are different biological agents which help in the process of pollination. Abiotic agents which help in the process of pollination are wind and water. Now why these abiotic agents? In nature, the flowers which are existing all are not colorful, all are not having some incentives to give to the agents for the process of pollination. Like for example nectar or the color of the flower and all. So those unattractive flowers, what will happen to them? So there are the abiotic agents which will be helping in the process of pollination. Now wind is one such abiotic agent. These type of pollen grains have a special kind of adaptations. They are very light in weight. They are large in number so that they can be easily carried by these agents. The aquatic plants, all the aquatic plants do not do the process of pollination by the help of water. There are some aquatic plants, say for example the common experiment which we are doing, Ecornia, or say for example lotus. Although they are aquatic, but in them the pollinating agent is not water. They are insect pollinated. But the plants like Hydrilla, Vallisneria, in them the water acts as an agent for pollination. So after pollination, the next step is fertilization. Fertilization, how does it happen? Perhaps you remember in the last class we have seen that inside the ovary this is the megasporangium. Now in the megasporangium, if you remember, that was 8 nucleate and 7 celled, isn't it? So when the pollen is transferred to the stigma, there is a dialogue exchange between the stigma and the pollen. Now dialogue exchange does not mean they are talking with each other. It is in terms of the chemicals. So when there is a self-compatibility, then only the pollen grain will germinate. It will pass through the style and it will go through this ovary, and finally it will reach to an end which is called the micropylar end. Now in this micropylar end is present the egg apparatus. The central one is the egg cell and these two are the synergids. Now this egg cell, before it enters the pollen grain, we have read in our previous class that they are shed in the two-celled condition. While they are shed in the two-cell condition, prior to its entry, that is your generative cell, it undergoes the second mitotic division, resulting into two haploid male gametes. One will come and fuse with the central egg cell here in the micropylar region, resulting into the formation of the embryo, and the second one will come in the central cell, that is the polar nuclei, and fuse with them to form a triploid cell, which is the endosperm nuclei. Now, this is the two times you see the fertilization is taking place. One time with the basal egg cell and another time with the central endosperm nucleus. Since two-time fertilization is taking place between the male and the female gamete, so we can call it to be a double fertilization, which is most common in case of most of the angiospermic plants. And this central cell is undergoing fusion between three nuclei, three haploid nuclei, these two, plus the second male nuclei, so it will be called as your triple fusion. So this is what happens in the stage of your fertilization. Now fertilization will result into formation of seeds, which will be of course the part of the post-fertilization stage of sexual reproduction in flowering plants. Now in case of the plants, if a plant or if a plant is undergoing continuously self-pollination, in that case sometimes it may lead to inbreeding depression. That means some negative qualities of the plant cannot be eliminated by regular practice of this self-pollination. So plants themselves naturally make their own devices, which are called as your outbreeding devices, to avoid this kind of self-pollination. Now, what are these outbreeding devices? Outbreeding devices means sometimes in some plants it has been seen that the same plant's pollen, even if it is mature and falls into the stigma, does not germinate. That is called your self-incompatibility. So, resulting into failure of fertilization. Sometimes you will see in some flowers, you have seen it in the morphology of flowering plants in class 11, that there is protogyny or protandry. That means sometimes the androecium is maturing faster than the gynoecium, sometimes the gynoecium is maturing faster than that of the androecium. So these are the natural processes inside the flower which will be preventing the process of self-pollination in case of flowers, and in that case they have to go for the other option, that is for the cross-pollination, to have a better result, to have a better quality characters in the next generation. Next, we will move on to artificial hybridization. Now, artificial hybridization, why we have to study? The reason is because, as you know, that India is an agriculture-based country. So, in that case, we have to think about improvement of food grains. So, for that, artificial hybridization is an important characteristic where we will be selecting the parents of desirable qualities, and we will be practicing the process of hybridization between them, so that we get a offspring of our own desire. So in that case, what we have to do is that at first we have to do the process which is called emasculation. Now, what is emasculation? Prior to the maturation of the anther, when we cut the anther, so that self-pollination does not take place. That is what is called as your emasculation. Now, it can be done by either cutting with the help of a scissor or some scalpel or something like that. Sometimes you can even dip the entire inflorescence into, hot water, but that should not be boiling water, of course. It has a certain temperature in which you can keep it, say 60 to 70 degrees Celsius, and then you can bag it. Now, when you bag the flower after cutting the anther or after doing this kind of treatment, then what happens is that there is less possibility of any other outside pollen grains to come and contaminate it, and you don't get the proper result of your hybridization experiment. So, when the stigma becomes mature, then you open it, and then you take a mature anther of the desired thing which you want to dust over it, and you dust it, and then again rebag it. So when you rebag it, it takes some time for the process of pollination, fertilization takes place, and that results into formation of seeds, and after some days when you open it, you get the desirable grains, or desirable, you can say, seeds, which can be stored or which can be grown to see whether you got the desirable characters in the offsprings or not. So this is how the artificial hybridization program is being conducted. Then next stage of this chapter, after the pre-fertilization is done, fertilization is done, next what? Next is called as your post-fertilization. So next is your post-fertilization. Now this post-fertilization, after the double fertilization which just now previously we have seen, then what happens is that this turns into to be the embryo and this one is the endosperm. Fine. Now soon after the fertilization process, the embryo does not go for further division. It takes some rest. In the meantime, the endosperm nucleus, it undergoes division, rapid division. Why does it go rapid division? Because it has to provide the nutrition to the developing embryo. So at first, after the fertilization, the first cell to undergo the division is your endosperm nucleus. But before it goes to the divisional stage, what happens is that the withering of the external parts takes place, like the anther will wither away, the calyx, corolla, all these things will be withering away. Now what will be happening is only inside the megasporangium, which is the embryo sac, where the various stages or the developmental things will be taking place. More or less in monocots and dicots, the developmental stages are all the same. In case of seed, if you see that when the seed formation will take place, you remember the nucellus? The nucellus turns out to be the integuments of the seeds, and that will become harder, or the seed coat, and the micropylar end, that will be represented by a small opening or a small pore through which water or exchange of gases can take place inside the seed. Now if you see a typical dicot seed, it has an embryonal axis. These are the two cotyledons and this is the embryonal axis. This portion of the embryonal axis is called as your epicotyl, and the tip of the epicotyl, this one is called as your plumule. And this portion to which it is joined to that of the cotyledon, this portion, this is called as your hypocotyl, and the tip of the hypocotyl will be called as your radicle. Now this radicle is again protected by that of your root cap, for the protection of the root to be penetrating deep into the soil for getting the nutrients and all. And it has been noticed that most of the dicot seeds, the endosperm is used up by this developing embryo for its development. So either the endosperm is absent or it is scarcely present within the that is your seed. Whereas if you see the monocot seeds, in case of the monocot seeds, if you see, in case of monocot seeds, the endosperm is not completely utilized. Endosperm is located and it is known as your scutellum in case of your monocot seeds. And in case of monocot seeds, if you see the structure of the monocot seeds, this is somewhat like this. And here will be the leaf primordia, which is called as your coleoptile, and this one is a root primordia, which is called as your coleorhiza. And this one is actually the scutellum or the endosperm. And this is one cotyledon, so that is why they are called as your monocots, and this is two cotyledon, that is why they are called as your dicots. So this is how the things goes inside the seed after the process of your fertilization. Now the seed soon after the formation does not germinate. It goes into dormancy. In some cases it has been noticed that seed can go to dormancy for even 10,000 years or 2,000 years. The oldest seed which has been found is your lupus, which is found from that of your Arctic tundra, which is 10,000 years old, and it has been successfully again germinated and it had flowered also. And similarly, 2,000 years old seed has been found, that is a kind of palm, date palm, Phoenix dactylifera, and that also has the viability of germination. So seed undergoes a period of dormancy, and when they get the suitable conditions for germinations, they can germinate even if it is many years. Okay, so this is about your seed germination and the seed dormancy. Now fruit formation. Now ovarian wall transforms into the outer wall of the fruit, which is called as your pericarp, and inside that fleshy part is called as your mesocarp, and the seed where it is just adjacent to the seeds, the layer is called as your endocarp. Now in case of fruit formation, if the fruit is formed from the ovary, then we call that type of fruit to be the true fruit. And if the other parts of the flower, say for example thalamus, it becomes fleshy and it takes part in the formation of the fruit. In that case, we call that type of fruit to be false fruit. Why we call them false fruit? Because here the ovary is not participating in the formation of fruit. Here some other parts of the flower other than ovary are taking part in the formation of fruit. So we call them as your false fruit. Example is your apple, strawberry, pear, etc. Parthenocarpic fruit. This is a third category of fruit, you can say, where there is no fertilization, no seed formation. Here the ovary transforms itself into the fruit. So here there is no seed. Ovary is taking part in the formation of fruit, but there is no fertilization. So that is why these type of fruits are called as your parthenocarpic fruit. Example is your seedless variety of fruit, like seedless grape, seedless banana, etc. Post-fertilization stage begins with that of seed formation. We have seen that. Now, seed, in case of dicot seed and monocot seed, the process of formation is almost same. That also we have discussed. The seed undergoes a period of dormancy, and after the period of dormancy, when it gets the right condition, then it again germinates. And we have seen that there are many seeds which are viable even for 10,000 years. What was the name? You remember the name? The name was Lupinas, isn't it? 10,000 years old, but it again germinated. Similarly, another very old seed we have talked about, that was your kind of date palm, that is Phoenix dactylifera. That was 2,000 years old. Now, next we are going to read about another type of seeds which is not as a result of fertilization. Sometimes in nature, seeds are also formed without fertilization. That type of seed is called as your apomictic seeds, and the phenomenon is known as your apomixis. So apomixis is a kind of asexual method of reproduction that results into formation of seeds, but it is not by the fusion of gametes. That means no syngamete takes place. So it mimics sexual reproduction. Why does it mimic sexual reproduction? Because the ultimate product of sexual reproduction is seeds. At the end of apomixis, we are getting the seeds. But here it is not purely a sexual reproduction because no fusion of male and female gamete takes place. So that is what is called as your apomixis. Now what happens in case of apomixis? This is an embryo sac. In case of embryo sac, if you remember, in the last day's class we have discussed that the functional megaspore mother cell, it undergoes meiosis. But in case of apomixis, inside the embryo sac, there is no meiosis, no reduction or division takes place. The functional megaspore cell, which is a diploid cell, it will result directly into the formation of the embryo. That means if it is suppose here and it is diploid, how the next stage of the embryo takes place inside the embryo sac, that division takes place resulting into the formation of your epicordyl, hypocordyl and the seeds and all those things, that will be totally same. But there is no fusion of the male and the female gamete, that is the egg cell. Here the meiosis does not take place. The megaspore mother cell, which is diploid, it is present inside the embryo sac. The further development of formation of embryo is same like that what happens in case of the normal natural kind of your sexual reproduction. But here there is no entrance of the male gamete to fertilize the female gamete, because here it is not a gamete. Here it is a diploid cell. Megaspore mother cell behaves directly as a that is your embryo cell, and it undergoes a further development. So this type of formation of seed is known as your apomixis. What is the advantage of these kinds of seeds of apomixis? That means suppose if we have the F1 generation of seeds as a result of hybridization, and if it has the property of apomixis, we can restore the parental characters which otherwise in normal conditions will segregate in the F2 generation. So apomixis is a kind of boon to the farmers, that they are able to restore the good qualities, the hybrid qualities of both the parents for generations to generations. So from agricultural point of view, apomixis seeds are very important. So scientists are thinking about a technique of just finding out those genes which are actually influencing this process of apomixis in the seeds, and they want to incorporate these genes in the crop plants also, so that apomictic seeds of the crop plants also can be harvested, and as a result of it we can do a much progress in terms of crop production. Now the last topic of our chapter, that is your polyembryony. Now what does this term polyembryony indicate? See here two terms are there, poly and embryony. Poly means many and embryony means embryos. Now let us look into this embryo sac. This is a typical dicot embryo sac. Inside this, as a result of normal sexual reproduction, this is the egg cell. When that your pollen tube will come here and one of the male gamete will fuse with it, fertilization will take place, result into formation of one embryo. That is a natural process, isn't it? But sometimes it so happens that the cells which are present in the nucellar region, now this is the nucellar region, you can see this is the nucellar region surrounding the embryo sac, and this one is the integumentary layers which is surrounding the nucellar cells. So sometimes the cells from the integuments or from the nucellar portion intrudes into the embryo sac, and these will be what? These will be the diploid cells, isn't it? These will be the diploid cells. So when they will be entering into, these are the diploid cells, they will start behaving as embryos, because the next developmental stages of these cells will be same like that of the developmental stages which will be shown by this one as a result of fusion between the male and the female gamete. This one as a result of normal or natural sexual reproduction, and this one the diploid cells of the nucellar that is a layer or that of your integumentary layers will be entering into, and they will now start multiplying in the same manner as the normal embryo. Now since so many cells will be behaving like that of the embryo, so inside that is your megasporangium, now so many that is your embryos will be formed, so we call it as your polyembryony. If you happen to have seen a seed of citrus fruit, say lemon or that of your orange, and if you squeeze it, you will find that inside so many small dotted type of structures are coming. Each one of them represents one embryo, and if you germinate them, then from a single seed you will see so many plantlets are germinating. So if a single seed is capable of giving so many plants at a time, so they will be having the same genetic constitution. So this is also important in terms of your agricultural practices. So polyembryony and apomixis are important in terms of increasing the agricultural product. So in this chapter, second chapter of sexual reproduction in flowering plants, we have ultimately seen that how does the plants show the process of gametogenesis by means of microsporogenesis and megasporogenesis. Then we have seen that how the zygote is formed as a result of fertilization, that is prior to this pollination and then fertilization. After fertilization, then next what? That is the post-fertilization stage, formation of seeds. That is how the developmental stage takes place in the formation of the dicot seed and the monocot seed, which is more or less the same. Endospermic seeds and non-endospermic seeds. That is, endospermic seeds will be having the endosperm intact in it, and non-endospermic seeds means the endosperm will be totally utilized by the embryo within the seed. So that will, most of the dicot seeds are non-endospermic and most of the monocots are the endospermic seeds. Then seed formation without fertilization, in that we have seen about the apomixis, where the formation of seeds takes place, but there is no sexual reproduction. So here the apomictic seeds are mimicking, they are copying sexual reproduction, but actually they are not undergoing the sexual reproduction to form the seeds. And finally polyembryony, in which inside the embryo sac, instead of one single embryo, too many embryos can form because of the intruding of the other diploid cells from the nucellar region or from the integumentary region. So this is all about the second chapter of sexual reproduction in flowering plants. I hope you have understood it well.